Complete the table by putting tick or cross in front of the activities that the items are able to carry out. Let us learn about the different life processes. Nutrition, respiration, excretion, transportation, reproduction, sensitivity, and growth are some of the processes that help the living being to sustain. These processes that are common to all living beings are called as the life processes. Various life processes work together and help in maintaining life. Let us study some of the life processes in plants and higher animals. Energy for life processes. All living organisms require energy to perform various activities. This energy is derived from the food they eat. Thus, food is a kind of fuel which provides energy to all the living organisms. Food contains several components called as nutrients. Based on their functions, nutrients are divided into three types: energy-giving nutrients, carbohydrates, fats; body-building nutrients, proteins; protective and regulating nutrients, vitamins and minerals. On the basis of chemical nature, the nutrients are of two types. Inorganic nutrients. Plants get their nutrients from inorganic substances present in the soil and atmosphere through leaves and roots. They convert it into organic compounds through the process of photosynthesis. These compounds are used for growth and to form various parts of the plant. Organic nutrients. They include carbohydrates, proteins, and fats which are of animal and plant origin. Nutrition. The process of intake of nutrients and its utilization by an organism is nutrition. Nutrition provides the raw materials needed to sustain the life. The way by which an organism obtains its food is the mode of nutrition. Using the hint, guess who we are. We prepare our food by using simple inorganic substances. We are plants. We use complex substances, break them up into simple ones, and use them for upkeep and growth. We are animals. Autotrophic nutrition. Autotrophic nutrition is the mode of nutrition in which organisms synthesize their own organic food. They use simple inorganic substances present in the surroundings for this. Such organisms are called as autotrophs. Most of the plants are autotrophs. Heterotrophic nutrition. Organisms that depend on other organisms for their food are heterotrophic organisms. All animals and fungi are heterotrophs. Different organisms use different strategies to obtain food depending upon their body design, organization, functioning, and also availability of food material. Fungi like yeast, mushrooms, and some carnivorous plants break down the food material into simple substances outside the body and then absorb it. Some organisms take in whole food and then break it down to simple substances inside their bodies and then absorb it. Example, human beings, cat, cow, etc. Some animals and plants derive nutrition from other plants or animals without killing them, but during the process harm the host plant or animal. Example, cuscuta, ticks, lice, leeches, tapeworms, etc. As the complexity of organisms increases, some of the organs of the body become specialized to perform specific functions. These organs together constitute a complex digestive system. Yet, the following basic steps of nutrition remain the same in all animals. Let us study about nutrition in human beings. All the processes involved in nutrition in human beings take place by association of digestive organs called the digestive system. The digestive system includes alimentary canal and the digestive glands. Alimentary canal is a long and muscular tube of varying diameter which extends from mouth to anus. The salivary glands, liver, the pancreas, and the gastric glands are the digestive glands associated with the alimentary canal. Let us perform this activity. Take 1 milliliter of starch solution, 1% in 2 test tubes A and B. Add 1 milliliter of saliva to test tube A. Leave both the test tubes undisturbed for 20 to 30 minutes. Now add a few drops of dilute iodine solution to the test tubes. What is the color change in test tube A and test tube B? What conclusion can you draw about presence of starch? We can conclude that our food contains starch. What does this tell us about the action of saliva on starch? Saliva digests the starch present in the food. Now let us study about the organs of digestive system in human beings. Mouth. The alimentary canal begins with mouth. We eat a variety of food items which have to pass through the same digestive tract. Naturally, the food has to be processed to generate particles with small size. Such crushed food is wetted with saliva secreted by the salivary glands so that the food can smoothly pass through the soft lining of the alimentary canal. The food that we take is of complex nature. It is converted into simpler molecules with the help of biological catalysts called as enzymes. Enzyme salivary amylase breaks down starch into a simple sugar maltose. Thus, digestion starts in the mouth itself. Stomach. The partly digested food goes down through the esophagus into a J-shaped stomach placed on the left side of the abdomen. The food is pushed forward in the canal due to rhythmic contraction and relaxation of the muscles of the canal called as peristaltic movement. The gastric glands present in the wall of the stomach release 3 gastric juices, namely hydrochloric acid, enzyme pepsin, and mucus. Hydrochloric acid creates acidic conditions which are necessary for the action of the enzyme pepsin, which digests proteins. Mucus protects the inner lining of the stomach from the action of acids under normal conditions. Can you think of any other function served by hydrochloric acid? You must have heard people complaining of acidity. Is it related to the hydrochloric acid present in the stomach? Hydrochloric acid lowers the stomach pH. If the pH of the stomach increases, the acidity in the stomach increases. The muscular walls of the stomach churn the food and help in proper mixing of the food with gastric juices. The exit of the food from the stomach is regulated by the sphincter muscle, which release small amount of partly digested food into the small intestine. Small intestine. The small intestine, about 5 to 6 meter, is the longest part of the alimentary canal fitted into a compact space in the belly by extensive coiling. Although it is much longer than the large intestine, it gets its name from its comparatively smaller diameter. The length of small intestine differs in various animals depending on the food they eat. Carbohydrates, fats, and proteins are fully digested in the small intestine. Since this action takes place only in alkaline conditions, the acidic food received from the stomach has to be first made alkaline. Liver, the largest gland in the body, secretes bile juice, which is stored in the gallbladder. Whenever food enters the small intestine, the gallbladder releases bile into it through a duct. Bile makes the food alkaline and breaks the large fat globules into smaller ones. This increases the enzyme action. Pancreas. Pancreas secretes pancreatic juice. It has digestive enzymes like trypsin to digest proteins, lipase to break down fats, and pancreatic amylase to digest carbohydrates. The bile and the pancreatic juice enter the small intestine through a common duct. Various intestinal juices are also secreted by walls of the small intestine to complete the digestion process of converting proteins to amino acids, complex carbohydrates to glucose, and fats into fatty acids. The digested food is absorbed by the walls of the small intestine, having numerous finger-like projections called villi. Villi are richly supplied with blood vessels which absorb the food. The absorbed food is taken to each and every cell of the body where it is utilized for obtaining energy. Unused glucose is stored in the liver in the form of glycogen. The unabsorbed food is sent to the large intestine. Large intestine. The undigested food and the residue of the digested food pass into the large intestine. It is about 1.5 meter long. This part of the intestine does not have any digestive function as the digestion is already completed. The walls of the large intestine have villi which absorb most of the water and salts. The rest of the material is removed from the body through the anus. Here too, the exit is regulated by the muscles called sphincter. Let us study about the nutrition in plants. Like animals, plants also need food for growth and energy. Green plants synthesize their food during the process of photosynthesis. They take in carbon dioxide and water and convert them into carbohydrates, glucose, in the presence of chlorophyll and sunlight. The carbohydrates are used for providing energy. The unutilized carbohydrates are stored in the form of starch. Let us look at the process of photosynthesis. Carbon dioxide enters into the leaves through tiny pores present on the surface of the leaf called stomata. Water is taken up from the soil. Chlorophyll, present in the chloroplasts, absorbs the radiant energy from the sunlight. It creates such conditions that light energy splits up the water molecules into hydrogen and oxygen, releasing energy in the form of adenosine triphosphate, ATP. The energy produced is used to reduce carbon dioxide to carbohydrates. The above steps may or may not take place one after another immediately. Example, desert plants take up carbon dioxide at night and prepare an intermediate product which is acted upon during the day by the energy absorbed by chlorophyll. What is the site of photosynthesis? The process of photosynthesis takes place only in green plants, mostly in the leaves, and to a lesser extent in green stems and floral parts. Look at this activity. Observe the given diagram of cross-section of a leaf as seen under the microscope. In which layer must the process of photosynthesis be taking place to a greater extent, and why? The photosynthesis must be taking place in the mesophyll cells because these cells contain more chlorophyll. Think over it. What is the significance of waxy cuticle on the upper and lower surface of a leaf? Waxy cuticle on the upper and lower surface of a leaf help to reduce loss of water from the leaf. Let us see what are the raw materials required for photosynthesis. Let us perform this activity. Take a potted croton plant. Keep the plant in a dark room for 3 days so that all the starch gets used up. Now keep the plant in sunlight for about 6 hours. Pluck a leaf of the plant and mark the green areas on it. Dip the leaf in boiling water for a few minutes. Now Immerse the leaf in a beaker containing alcohol. Carefully place the above beaker in a water bath and heat the alcohol till it begins to boil. What happens to the color of the leaf? The green parts of the leaf become decolorized. What is the color of the solution? The solution turns green in color. Now immerse the leaf in a dilute solution of iodine for a few minutes. Take out the leaf and rinse off the iodine solution. Observe the color of the leaf, especially the marked regions. What do you conclude? We can conclude that photosynthesis takes place only in the presence of chlorophyll. There is an absence of starch in the parts of the leaf which are devoid of chlorophyll. Let us look at one more activity. Take two healthy potted plants which are nearly of the same size. Keep the plants in dark for 3 days and destarch them. Now place each plant on a separate glass plate and place a watch glass containing potassium hydroxide by the side of one of the plants. Potassium hydroxide is used to absorb the carbon dioxide. Cover both the plants with separate bell jars. Use Vaseline to seal the bottom of the jars to make the setup airtight. Keep the plants in sunlight for 2 hours. Pluck a leaf from each plant and check for the presence of starch. What can you conclude? We can conclude that without carbon dioxide, photosynthesis cannot take place. There is absence of starch in the leaves where the air from the jar did not contain any carbon dioxide. Now let us do the activity to demonstrate that sunlight is essential for photosynthesis. 1. We take a potted plant having green leaves and place it in a completely dark place for about 3 days to destarch its leaves. So in the beginning of the experiment, the leaves do not have any starch in them. 2. Take a thin strip of aluminium foil and wrap it in the center of one leaf on both the sides while the leaf is still attached to the plant. Aluminium foil should be fixed tightly to the leaf by using paper clips so that sunlight may not enter it from the sides. The aluminium foil should cover only a small part of the leaf so that the remaining part of the leaf remains uncovered and exposed to sunlight. We have covered the center part of the leaf with aluminium foil so that sunlight may not fall on this covered part of the leaf. 3. Keep this potted plant with partially covered leaf in bright sunlight for 3 to 4 days. 4. Pluck the partially covered leaf from the plant and remove its aluminium foil. This whole leaf is now to be tested for the presence of starch. But before testing for starch, chlorophyll has to be removed from the leaf. This is because chlorophyll interferes in the test for starch due to its green color. 5. Put the plucked leaf in a beaker containing some alcohol. Place the beaker containing alcohol and leaf in a water bath. The water bath is being used here for heating alcohol because alcohol is a very inflammable liquid. So if alcohol is heated directly over a flame, then it will catch fire at once. 6. Heat the water in the bigger beaker. Then the alcohol in the smaller beaker will also get heated and start boiling soon. This boiling alcohol will extract chlorophyll from the green leaf. 7. Boil the green leaf in alcohol till all its green pigment, chlorophyll, is removed. The leaf will now become almost colorless or pale, and the alcohol will turn green. 8. Remove the colorless leaf from alcohol and wash it thoroughly with water to remain any chlorophyll which may be sticking to it. 9. Place the colorless leaf in a petri dish. Drop iodine solution over the decolorized leaf with the help of a dropper. Observe the change in color of leaf. 10. The middle part of leaf, which was covered with aluminium foil, does not turn blue-black on adding iodine solution, showing that no starch is present in this middle part of the leaf. This is because sunlight could not reach the covered middle part of the leaf, due to which the covered middle part of the leaf could not do photosynthesis to make starch. 11. The uncovered part of leaf on both sides of the aluminium foil, which was exposed to sunlight, turns blue-black on adding iodine solution, showing that starch is present in this part of leaf. This means that the part of leaf which was exposed to sunlight could do photosynthesis to make starch. 12. Since the part of leaf which was covered and hidden from sunlight does not contain starch, but the part of the leaf which was exposed to sunlight contains starch, therefore we conclude that sunlight is necessary for photosynthesis to make food like starch. From the above experiment, we actually get two conclusions, that A. Sunlight is necessary for the process of photosynthesis, and B. Leaves make starch as food by photosynthesis. Raw materials of photosynthesis are carbon dioxide and water. With the help of sunlight, chlorophyll, water and carbon dioxide, the raw materials taken in through the stomata of the leaves of autotrophs synthesize carbohydrates and meet their energy requirements. Along with this, they also need body-building and regulating nutrients like nitrogen, phosphorus, iron, magnesium, etc., which are taken up from the soil. Nitrogen is essential for synthesis of proteins. It is taken up in the inorganic form or in the form of an organic compound. Think over it. Can you recognize this plant? It is a pitcher plant. It is a heterotroph. It grows in the soil which has very less nitrogen. Find out how does this plant gain nitrogen. The trapped insects are converted into amino acids, phosphates, urea, from which the plant obtains its mineral nutrition, particularly nitrogen and phosphorus.